Hi, hello. This is Dr. Jagadish Narendra Prasad, Senior Lecturer in Botany, Sri Chaitanya Gosala, Vijayawada. Today, we are going to discuss about what is nucleotide, what is nucleoside, and what are nucleic acids, what is their role in living systems, all these things in detail. First, let us see about nucleotides. When it comes to the nucleotides, before discussing nucleotides, let us see the components of nucleotides. When it comes to the components of nucleotides, nucleotides include a nitrogen base, pentose sugar, and phosphate. Nitrogen bases includes adenine, guanine, thymine, cytosine, uracil. These nitrogen bases are heterocyclic compounds. So why they are called as heterocyclic compounds? Because they have a ring which is made with carbon and nitrogen. Here there is carbon, nitrogen, carbon, nitrogen, carbon, nitrogen, carbon, nitrogen. So as the ring is made with the CN, carbon and nitrogen, so these are said to be heterocyclic compounds. Adenine is one kind of nitrogen base, guanine is other kind of nitrogen base. These two are said to be substituted purines. Whereas cytosine, thymine and uracil are substituted pyrimidines. Substituted pyrimidines. Purines are made with the dicyclic rings, that they have two cycles in their structure, a 6 atom and 5 atom structure, whereas pyrimidines are monocyclic. That is the importance of purines and pyrimidines. So purines includes adenine and guanine, pyrimidines include thymine, cytosine and uracil. Apart from nitrogen base, a nucleotide also contains sugars. These are also called as pentose sugars. These pentose sugars are of two kinds. One is ribose sugar and other one is deoxyribose sugar. The main difference exists between ribose sugar and deoxyribose sugar is at 2' carbon. If you take, this is the cycle of a pentose sugar, it is considered to be 1' carbon, 2' carbon, 3' carbon, 4' carbon and 5' carbon. Similarly, here also say 1', 2', 3', 4' and 5'. At 2' position, there is a HOH in case of ribose, whereas in case of deoxyribose, there is a H. There is no hydroxyl group at the 2' position. Therefore, this makes the difference between ribose and deoxyribose. Deoxy, the name itself is indicating that one oxygen is lacking in this. Ribose sugar is a component of RNA nucleotides. Deoxyribose sugar is a component of DNA nucleotides. What is DNA? Deoxyribonucleic acid. This nucleic acid is called as deoxyribonucleic acid as they have deoxyribose sugar in their structure. Whereas RNA is ribonucleic acid. Ribonucleic acid because it has ribose sugar in its structure. And another most important thing, phosphoric acid or phosphate is also present in nucleotides. When nitrogen base joins with pentose sugar, it gives nucleosides. When nucleoside joins with phosphate or phosphoric acid, then it gives nucleotide. That is the difference between nucleoside and nucleotide. Simply to say, in nucleotide there is a phosphate. If phosphate is removed from the nucleotide, you will be getting nucleoside. If you take RNA nucleosides, adenosine, guanosine, cytidine, uridine, or adenosine nucleosides. Whereas DNA nucleosides, these are RNA nucleosides. Whereas DNA nucleosides, I am saying, if you take the DNA nucleosides, it is deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine, or simply you can also call it as thymidine. That is the difference between nucleosides of DNA and RNA. The only difference that you can find here is there is deoxyribose sugar to which nitrogen base is attached here, whereas here nitrogen base is attached to ribose sugar. When these nucleosides, when phosphate is added to these nucleosides, then you will be getting nucleotide. Phosphate is attached at 5' carbon of pentose sugar. At this position, phosphate is added. If phosphate is added, then it is called a nucleotide. The nucleotides of DNA, of RNA includes adenylate acid, guanylic acid, cytidilic acid, and uridylic acid. These are nucleotides of RNA. Whereas nucleotides of DNA, deoxyadenylate acid, deoxyguanylic acid, deoxycytidilic acid, and thymidilic acid. There is no uracil in DNA. There is no thymine in RNA. RNA has uracil, DNA has thymine. That is the difference. When deoxyribose sugar is attached, look at here, the difference between nucleotides and nucleosides. In a nucleotide, nitrogen base is attached to the 1' carbon of the pentose sugar, and phosphate is attached to the 5' carbon of the pentose sugar. Whereas in nucleoside, nitrogen base is attached to the 1' carbon and there is no phosphoric acid. This is the difference between nucleoside and nucleotide. So, if phosphate group is also found esterified to the sugar of a nucleoside, then they are called as nucleotides. As I said, adenylate acid, thymidilic acid, guanylic acid, uridylic acid, cytidilic acid are nucleotides. And nucleic acids includes DNA and RNA. These are the nucleic acids. So, a nucleotide contains nitrogen base, pentose sugar, and phosphoric acid. Nitrogen bases, again two kinds, purines, pyrimidines. Purines include adenine, guanine. Pyrimidine include thymine, cytosine, uracil. Uracil is present only in case of RNA. Thymine is present only in case of DNA. Uracil is not seen in DNA. Thymine is not seen in RNA. Among the pentose sugars, deoxyribose sugar is present in DNA. Ribose sugar is present in RNA. Ribose sugar is present in RNA. That is the difference between these two. And nucleic acid along with nucleic acids are also heteropolymers. These are heteropolymers. They are said to be polynucleotide chains because the building blocks of nucleic acids is nucleotide. This nucleic acid along with polysaccharides and proteins, they constitutes true macromolecular fraction, and they got separated into acid insoluble pool of the living system. Acid insoluble pool. They are not separated into acid soluble pool because they are macromolecules. This is very, 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 very important. And I said these nucleic acids are polynucleotide chains, that they are made with many nucleotides. So building blocks of the nucleic acids is nucleotides. And nucleotide contain three chemically distinct components. What are they? One is a heterocyclic compound, and the second is a monosaccharide, and third is a phosphoric acid or phosphate. The heterocyclic compounds are purines and pyrimidines. So what are purines? Adenine and guanine. Pyrimidines, thymine, cytosine, uracil. These are the pyrimidines. Monosaccharides. Monosaccharide is nothing but it is a pentose sugar. Pentose sugar like ribose sugar and deoxyribose sugar. And phosphoric acid is phosphate is also present in the nucleotides. Now, the another most important thing is, as we discussed, deoxyribose sugar is present in DNA and ribose sugar is present in RNA. The most another important thing, nucleic acids containing deoxyribose sugar is DNA and nucleic acid containing ribose sugar is RNA. And these two are nucleic acids. In this, both can act like a genetic materials. DNA can act like a genetic material. RNA can also act like a genetic material. But most of the organisms have DNA as genetic material. All cellular organisms, including all prokaryotes and all eukaryotes, have DNA as genetic material. Exceptionally, some viruses, some plant viruses and some plant animal viruses have RNA as genetic material. If you take DNA, DNA has got double helical structure. This double helical structure was proposed by Watson and Crick. So this is also called as Watson and Crick model. And the model proposed by Watson and Crick is called B type of DNA. And it is one kind of secondary structure. DNA is composed of two strands, two helically coiled strands. Two helically coiled strands. These two strands anti-parallel to each other, which means the polarity of one strand is 5' to 3'. The polarity of one strand is 5' to 3'. Then the polarity of opposite strand is 3' to 5'. This is called anti-parallel. So the two strands are helically coiled and they have anti-parallel polarity. That's very, very, very important. Now, if you take the backbone of the DNA, the backbone is composed of sugar, phosphate, sugar. This is sugar, phosphate, sugar. This is one chain and this is another chain. In this chain, the backbone is again made with sugar, phosphate, sugar. In this, bases are projected perpendicular to the backbone. This is the backbone. Sugar, phosphate, sugar. And these are the nitrogen bases. Nitrogen bases are projected perpendicular to the backbone and projecting towards inside. Projecting towards inside. Similarly, here also same. This is backbone. Now these are the nitrogen bases. And another important thing, in a double-stranded DNA molecule, adenine always pairs up with the thymine or vice versa. Thymine pairs with adenine. And guanine always pairs up with cytosine or cytosine pairs with guanine. Never adenine pairs with guanine or cytosine, or thymine never pairs with cytosine or guanine. Only adenine can pair with thymine and only cytosine can pair with guanine. That is most important pairing. This itself is called as base pairing. And another most important thing, adenine pairs with thymine by 2 hydrogen bonds. This is 1 hydrogen bond and this is another hydrogen bond. 2 hydrogen bonds. Guanine pairs with cytosine by 3 hydrogen bonds. 3 hydrogen bonds. So, in a nucleic acid, nitrogen bases of adjacent chains paired by hydrogen bonding. Now, these are hydrogen bonds. This nitrogen base linked to pentose sugar by N-glycosidic bond. And phosphate is attached to the pentose sugar by ester bond or phosphoester bond. In a nucleotide chain, the succeeding nucleotides are linked by phosphodiester bond. This is very important and you have to follow very carefully that nucleotides of one strand are linked by phosphodiester bond. So why is it called as phosphodiester bond? Let us see now. This phosphate links to the succeeding nucleotides by 2 ester bonds on either side. Hydroxyl group of pentose sugar forms a ester bond with phosphate. Hydroxyl group present at 3' position of the pentose sugar forms a ester bond with phosphate, and this phosphate also forms a ester bond to the 5' carbon of next pentose sugar. That means phosphate is having 2 ester bond on either side, one towards one side and other one is towards other side. As there are 2 ester bonds on either side of the phosphate, this is called phosphodiester bond. This is one phosphoester bond and this is another phosphoester bond, together called as phosphodiester bond. So, phosphodiester bond formation takes place between 3' hydroxyl group. This is 3' hydroxyl group of pentose sugar of one nucleotide to the phosphate and to the 5' carbon of the pentose sugar of the next nucleotide. This phosphate is forming a ester bond with this pentose sugar and also with this pentose sugar. Hence, it is called as phosphodiester bond. So, in a nucleic acid like DNA, you could see 3 kinds of bonds. One is hydrogen bond, second one is N-glycosidic bond, and third one is phosphodiester bonds. Hydrogen bonds are present between adjacent nucleotides. N-glycosidic bond is present between nitrogen base and pentose sugar. And phosphodiester bonds are present between succeeding nucleotides. Now, bases of one strand always pairs with bases of other strand. How are they pairing? Like thymine pairs with adenine by 2 hydrogen bonds. Guanine pairs with cytosine by 3 hydrogen bonds. And another most important thing, the 2 strands are helically coiled so that they forms helices or helix. In a helix, the angle between 2 successive base pairs is 36 degrees. Now this is one helix. The pitch of the helix is 34 angstroms or 3.4 nanometers, and it has an angle of 360 degrees, because say if it is adenine, this is thymine, guanine, cytosine, cytosine, guanine, thymine, adenine, adenine, thymine, double bond, triple bond, triple bond, double bond. Now there are 5 base pairs in this 180 degrees of angle and another 5 base pairs in another 180 degrees of angle. Now the angle between two successive base pairs, between these two it is 36 degrees angle. The total helix has 360 degrees of angle and raised per base pair 3.4 angstroms, like between two successive base pairs the distance is 3.4 angstroms. So that the total helix will have 34 angstroms of length or pitch. Therefore, this helix has 10 base pairs or 20 nucleotides. 10 base pairs or 20 nucleotides. 10 in one strand and 10 in other strand. So as we discussed earlier, there are 10 purines in a helix and 10 pyrimidines in a helix. 10 purines and 10 pyrimidines. Therefore, 20 dicyclic rings and 10 monocyclic rings will be present. So, this kind of structure of DNA itself is called B type of DNA, and it was proposed by Watson and Crick. These are the few important details regarding the nucleic acids. We will discuss much more details in the next coming videos. Thank you.